Hello students, my name is Niyati Said and thanks for watching Wikipedia word videos. My topic for the presentation is plant growth regulators students. In this section of the video, we will discuss about the plant hormones which we commonly called as plant growth regulators. Plant growth regulators, they are small. They are simple molecules of diverse chemical composition, that regulate plant growth. Plant growth regulators, they could be indole compounds such as indole 3-acetic acid. They could be adenine derivatives, such as N6-perfluoryl aminopurine. They could be derivatives of carotenoids such as abscisic acid. They could be terpenes such as gibberellic acid. They could be gases too, such as ethylene. So plant growth regulators, they are variously described as plant growth substances. They are commonly called as plant hormone or phytohormones in literature. So based on the function, they are broadly divided into two categories. First is plant growth promoters and second is plant growth inhibitors students. This division is based on their function in a living plant body. So one group of PGR, that is plant growth regulator, they are involved in growth promoting activities. That's why it is called as plant growth promoters, such as cell division, such as cell enlargement, such as tropic growth. Tropic growth means growth towards the light or growth towards the gravity. So these are the tropic growths. Pattern formation, flowering, fruiting and seed formation, it all comes under plant growth promoters. So one group of plant growth regulator is plant growth promoter for growth promoting activities like cell division, cell enlargement, tropic growth, pattern formation, flowering, fruiting and seed formation. For example, Auxin, Gibberelins and Cytokinins. These are the plant growth promoters. These are also called as phytohormones students because these are the hormones of plants students. Second group of plant growth regulator is plant growth inhibitor. They play an important role in plant response to wound or any stress of biotic and abiotic origin. They are also involved in various growth inhibiting activities, like dormancy, like abscission, and more importantly, they respond to the wound and stresses of biotic and abiotic origin. The PGR, that is plant growth regulator, if we talk about the plant growth inhibitor to be more precise, they belong to this group. Abscisic acid, it belongs to this group. Inhibitor. Ethylene also belongs to the plant growth inhibitor group. So this is gaseous plant growth inhibitor, ethylene. It could fit either of the group, but it is largely an inhibitor of growth activities. So keep this in mind that ethylene is a gaseous hormone and it could fit either of these two groups. First is plant growth promoter and second is plant growth inhibitor. But ethylene largely fits a growth inhibitor group, that is plant growth inhibitors. I hope it is clear to you now that plant growth regulators, they are small, they are simple molecules, but they have diverse chemical composition. And commonly they are called as phytohormones or plant hormones. Plant growth regulators, they are divided into two major groups. First is plant growth promoter and second is plant growth inhibitors students. Plant growth promoter for growth promoting activities like cell division, cell enlargement, tropic growth, pattern formation, flowering, fruiting and seed formation, they all come under plant growth promoters. For example, Auxin, Gibberelin and Cytokinin, they are the examples of plant growth promoters. Now come to the plant growth inhibitors. They are those regulators that inhibit activities like dormancy, like abscission, and they, more importantly, they respond to wound and stress of biotic and abiotic origin. Examples are abscisic acid and ethylene. And please note that this ethylene, it fit either of the group, but it largely fits growth inhibitor group. Now we will discuss about the auxin. Auxin is a phytohormone, or you can say it is a plant hormone, students. Interestingly, the discovery of each of the 5 major groups of plant growth regulators have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin. Charles Darwin and son Francis Darwin, he started observation when they observed that coleoptile of canary grass responded to unilateral elimination by growing towards the light source. See, this is growing towards the light source. This is the coleoptile of canary grass. And in the one direction, that is unilateral direction, we are eliminating with the light source. After a series of experiments, it was concluded that tip of coleoptile, see, this is coleoptile of canary grass and this is the tip of the coleoptile of the canary grass. It is concluded that tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. See, this coleoptile has bent it towards the light source. Auxin was isolated by W. Wendt from tips of coleoptile of oat seedling. So, Charles Darwin and his son Francis Darwin, he observed that coleoptile of canary grass, it responded to the unilateral elimination by growing towards the light source. This movement, we call it as tropic movement, and to be more precise, it is phototropism. Photo means light and tropism means growth. So growth of a plant towards the light is known as phototropism. It was concluded that the tip of the coleoptile caused the bending of the entire coleoptile. See, this is the coleoptile of a canary grass. This is the light which we are eliminating, and this is the source of the light, and this coleoptile has bended towards the source of the light. This shows that the tip of the coleoptile caused the bending of the entire coleoptile. F.W. Wendt, he isolated auxin, which is a plant hormone, from the tip of the coleoptile. See, this is the tip of the coleoptile, this green, okay? And F.W. Wendt, he was a scientist, and he isolated auxin from the tip of the coleoptile of oat seedling. Auxin was first isolated from human urine. Auxin was not isolated from coleoptile tip first, okay? It was first isolated from human urine. So, a question may be asked, like, human urine contains auxin or not? So, yes, it contains auxin. Auxin is the only hormone which is found in human urine, students. Auxins, they are produced by growing apices of stems and root, from where they migrate to the region of their action. Okay. Now, discovery of the auxin experiment of Boysen and Jensen. Wendt, in year 1928 from Holland, demonstrated the existence of a substance in the tip of oat, that is avena seedling, which diffused downward and promoted growth. He demonstrated that when the tip of the oat coleoptile was removed, growth stopped. That means auxin was there in the tip of the oat coleoptile, right? Which is responsible for the growth. So this way he discovered auxin, that is a plant hormone. Now, when a freshly cut coleoptile tip was placed on an agar block, okay, agar block for few hours so as to allow the growth substances to diffuse into, and agar block was then placed on the cut end of the coleoptile. Growth appeared. I'm explaining you here. This is the diagrammatic representation of this experiment, which was performed by Boyson and Jensen, who discovered Auxin. As I just taught you, that F.W. Wendt from Holland demonstrated the existence of a substance in the tip of oat seedling which diffused downward and promoted growth was Auxin, right? See, this is the coleoptile tip which is intact. In this, Auxin was there at the tip. But now, in the second phase, coleoptile tip is removed. See, this is removed here. And this tip, this is pink in color, and it is placed in the agar, and it is cut into blocks. See, this is cut into blocks. Now, freshly cut coleoptile tip was placed on the agar block for few hours because agar has growth substances. So, it will diffuse into the tip of the coleoptile, and agar block was then placed on the cut end of the coleoptile. See, this is the cut end of the coleoptile. Now, this agar block is again put into this cut end. So, what happened? Curvature occurs and beneath blocks. So, this shows the discovery of Auxin, which is responsible for the growth. Oat coleoptile experiments, let's see in detail. If agar block was placed laterally on the cut end of the coleoptile, only that side of the coleoptile elongated, resulting in curvature. See, this is the source of the light. This is the unilateral illumination for on distribution of the Auxin in the coleoptile. This is the zone of the elongation. This is the degradation of the Auxin by enzyme which is taking place here. This is the movement of Auxin. Auxin moves laterally away from the light. See, it is moving laterally away from the light. It is not greater concentration of Auxin on the shaded side eliminates cell elongation, thereby causing curvature. That's why it moves or it bends like this. Okay, so if the agar block was placed laterally on the cut end of the coleoptile, only that side of the coleoptile elongated, results in curvature. See, this is the curvature it is showing. The side of the coleoptile that received the growth substance elongated faster, and it caused the curvature towards the opposite side. He called this substance Auxin from the oat coleoptile experiment. Now let's see Auxin in detail. Type of Auxins. In this lesson, you will learn about the hormone Auxin and type of it that stimulates plant growth in response to the environmental cue. So what are Auxin in plants, students? If you watch a plant develop over a time, you will notice that it grows up, out and down. The stem grows up, right? See, this is the stem that is growing up over a time. You will notice that it grows up, out, down. A stem specifically, it grows up and it makes the plant taller. Leaf, see, these are the leaves that grows outward. See, it has grown outward. It reaches towards sunlight. The root, it grows downward, absorbing nutrients, and thus it provides stability from the ground. Now, what are the signals that parts of a plant to grow like this? Just like your body, plants have a variety of hormones, and auxin are a specific group of hormone that makes plant cell grow. Okay? So, auxin, they are not only critical to plant growth, but they were also the first class of the plant hormone to be discovered. As I just taught you that a Dutch graduate student whose name was F.W. Wendt, he described Auxin in year 1928, and he chose the name Auxin from a Greek word Auxin, which means to grow. So the main function of Auxin is to help plants grow. Auxin, it stimulates plant cells to elongate, and apical meristem of a plant is one of the main places that auxin produced. This makes sense because the apical meristem is also the location that all other parts of a plant grow, from stem, leaves and flowers. So type of auxins are two. First is natural and second is synthetic. Natural examples are indole 3-acetic acid, which we commonly call it as IAA. Okay, I for indole, A for acetic and A for acid. And second is indole butyric acid, which we commonly call it as IBA. They are isolated from plants. And second category of auxin is synthetic. For example, NAA, naphthalene acetic acid, and 2,4-D. Remember the student, 2,4-D is very commonly used auxin these days. 2,4-D, that is 2,4-dichlorophenoxyacetic acid. And these are the structures of natural as well as synthetic auxins. Okay. Now, what are the functions of auxin? We very well know that the main function of auxin is to help plants grow. Auxin, it stimulates plant cells to elongate, and the apical meristem of a plant is one of the main places that auxin is produced. See, this is the apical meristem of a plant. So auxin, it stimulates plant cells to elongate, and the apical meristem of a plant is one of the main places that auxin is produced. So this makes sense because the apical meristem is also the location that all other parts of the plant grow, from the stem, leaves and flowers, students. So what are the functions? It initiates rooting in the stem cutting for plant propagation. It promotes flowering. For example, in pineapple, it prevents fruit and leaf drop at early stage. It promotes abscission of older leaves and fruits. It induces parthenocarpy, such as seedless plants. I'll explain what parthenocarpy is in detail, but for the time being, just know that auxin is a plant hormone that induces parthenocarpy, for example, in tomatoes. Sixth function, that they are used as herbicides. That means it kills unwanted herbs. 2,4-D is a synthetic auxin which is used to kill dicot weeds. It does not affect monocot plant. It is used to prepare weed-free lawns. So a question may ask, which plant hormone is required to prepare weed-free lawn? So the answer is auxin, students. It controls xylem differentiation and it helps in cell division also, students. I hope it is clear to you now. See, this is the cut off tip of a plant where auxin stays. And then add auxin at the top. So these are the three things which could happen. Do nothing, or put tip back, or add auxin at the top. See, this is another where we add auxin at the base. Here this blue is representing auxin, and thus it induces rooting, or it induces roots in the stem cutting for the plant propagation. As I told you, it induces parthenocarpy. Okay, so this is a strawberry where we remove seeds. No addition, add auxin, so it enhances or it grows like this. Whereas if you don't add auxin, then it stays or it shows a stunted growth. So it promotes flowering also. In higher plants, the growing apical bud. See, this is the apical bud. Apical bud is a bud which is present at the top. That bud we call it as apical bud. So apical bud, it inhibits growth of lateral buds. See, these are the lateral buds. So this apical bud, it inhibits the growth of axillary bud. It is known as apical dominance. That means apical bud shows dominance over lateral bud. Removal of shoot tip, that we call it as capitation, it results in the growth of lateral bud. Obviously, when you cut apical bud from a plant body, then it will result in a growth of a lateral bud. It is applied in a tea plantation, which we call it as hedge making. See, this is the auxin which is present at the tip of a plant. Obviously, when auxin is present at the tip of a plant, then it shows apical dominance. That means it does not allow a lateral or axillary bud growth. So when auxin is present at the tip, then axillary bud, it remains in the dormant position. But when it is decapitated, that means when it is cut, then it shows outgrowth. This axillary bud, which was earlier at the dormant stage, then it will show outgrowth after decapitation. That means when you cut the apical bud. So you can say that auxin, it shows apical dominance in plants. See, this is apical dominance. See, this is the lateral bud. So when you cut the apical bud, then lateral bud, it shows growth. So this shows that at the apical portion, auxin is there, which is responsible for the growth of apical bud. As I just told you that auxin, it induces apical dominance, where the apical bud suppress the growth of the lateral bud. Okay, so this is the removal of or decapitated position where removal of the apical bud has taken place, and this is the lateral bud, and this is the leaf. See, lateral bud or axillary bud, they both are same. Okay, it is present at the position between the stem and the leaf. So this is the lateral bud that becomes active and it produces own cytokinin. Cytokinin is also a plant hormone, which I'll explain in detail. So lateral bud, it becomes active and it produces own cytokinin, auxin and gibberellic acid and growth inhibitor if present inactivated. And so this is the possible involvement of a plant growth substance in apical dominance after removal of the apical bud. And this is the possible involvement of a plant growth substance in apical dominance in presence of apical bud. See, here apical bud is present and lateral bud is also present. So gradient of auxin and gibberellin from apical bud, which attracts cytokinin and food. So cytokinin is a plant hormone that is responsible for the cell growth or cell division. Okay, so here food, cytokinin and mineral salts and root to the apex takes place. That means from root, all cytokinin, hormone and mineral salts, food, it all goes from root to the apical portion, which regulates further growth. And this is the dormant lateral bud, because apical bud is present, so this remains in the dormant position with a low cytokinin content. Cytokinin, as I told you, it is responsible for the cell division. It is a plant hormone, and it may also possess a growth inhibitor, which we call it as abscisic acid. Okay, so this is the possible involvement of plant growth substance in apical dominance in presence of apical bud. Third function of auxin is that it induces parthenocopy. That means the formation of a seedless fruit without the act of fertilization. Okay, so this is the cluster of flower on tomato. This is unpollinated flower. Here no pollination takes place and it thus drops off. And this is unpollinated flower. When we apply auxin to the stigma, then initiation of fruit takes place, because you have applied auxin to the stigma. Stigma is a part of a female reproductive organ. Okay, stigma, style and ovary, these are the three reproductive organs of female, okay, which is present in the flower. So this shows parthenocopy, that auxin, it induces parthenocopy, that is the formation of seedless fruits without the act of fertilization, students. Now, inhibition of abscission. Obviously, auxin, as I told you, that it is responsible for the growth. So, it inhibits abscission also. Formation of an abscission layer at the base of the petiole or pedicel, it results in the shedding of leaves, flowers or fruits. Okay, but auxin, it inhibits such abscission as they prevent the formation of abscission layer. See, this is a plant which has so many fruits, and this is a plant which has less number of fruits. So that means this is a plant with a high number of auxin, which prevents the abscission, and thus it shows growth of fruits. And this is showing the less number of fruits. That means it has less number of auxin, and thus abscission is there. Auxin, it is also used as weedicide. As I have taught you that auxin, it is used to prepare weed free lawn. Okay, so many synthetic auxin, they are used as selective weed killers and herbicides. 2,4-D is a synthetic auxin, that is whose full form is 2,4-dichlorophenoxyacetic acid. It is used to destroy broad-leaved weeds. Okay, and it does not affect mature monocotyledonous plants. Please remember this. So this is a broad-leaved weed killed by 2,4-D spray. 2,4-D is a synthetic auxin, and this is a wheat or another grass not injured by 2,4-D spray. See, this is the spray of 2,4-D. Before spraying a beta naphthoxyacetic acid, and this is after spraying beta naphthoxyacetic acid. So this clearly indicates that many synthetic auxin, they are used as selective weed killers and herbicides. Okay, and more importantly, synthetic auxin, that is 2,4-D, is used to destroy broad-leaved weeds. Okay, but it does not affect mature monocotyledonous plants, so keep that in mind. So some of the uses of synthetic auxins are given below. First is fruiting, second is rooting, third is weeding, and fourth is storage. NAA, that is naphthalene acetic acid, it is a synthetic auxin, and indole butyric acid, that is IBA, they both are synthetic auxins. They help in natural or parthenocarpic fruit setting that increases crop yield in tomato, pepper, and figs. Okay, fig is a fruit. And as I told you, it also induces rooting in shoot culture plant. So it is important technique to reproduce genetically similar plants, especially ornamental plants such as money plant. Okay, the cuttings of a plant, they are dipped in rooting powders that contain two synthetic hormone or synthetic auxins such as NAA and IBA. This promotes root initiation and stimulates their development. And third is weedicide. For example, 2,4-D, that is 2,4-dichlorophenoxyacetic acid, and 2,3,6-trichlorobenzoic acid. They are used to kill dicot weed among monocot crop, as latter are unaffected. As I told you that monocot plants are not affected. For storage, synthetic auxin, which we call it as 2-methyl-4-chlorophenoxyacetic acid, that is commonly called as MCPA, it inhibits sprouting of the bud in potato, and hence it is used in their storage. Okay, so these four are the uses of synthetic auxin, such as fruiting, rooting, weeding, weed, and storage. So this was all about first plant hormone, which we call it as auxin. In my next section of the presentation, we will discuss about the gibberellin, which is second important plant hormone. So till then, stay tuned and keep watching Edupedia Word videos. Thank you.